Hello, everyone, and welcome to today's keynote webinar titled Gene Therapy Manufacturing. This webinar is part of the 5th annual event in the Bioprocessing Virtual Event Series, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our live chat feature during this presentation. You can find that live chat located at the left of your screen. You can also participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of this presentation. Now, if you have any trouble seeing or hearing this presentation, just simply click on that help desk button located at the top of your navigation bar or from the lobby of the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on that continuing education credits link located at the abstract tab from the menu at the left of your screen and follow the process to obtain those credits. I now present today's speaker, Dr. Baley Reeves, the interim director at the National Center of Therapeutics Manufacturing in Texas A&M Engineering. For a complete biography of our speakers, please visit the presenter tab from the menu at the left of your screen. Dr. Reeves, welcome. You may now begin your presentation.
Good morning, everyone. So today I'd like to go over a high-level overview about gene therapy manufacturing and how it kind of differs from traditional biologics manufacturing. So I'm coming to you from NCTM. NCTM stands for the National Center for Therapeutics Manufacturing. We are located on Texas A&M's campus in College Station, Texas, and we are primarily a workforce development organization focusing on everything from traditional biologics to vaccines to advanced therapies. We have a simulated cGMP facility for training. So we really want to try to make everything manufacturing relevant. So we have a mock production facility, pilot scale equipment, and the way that we run our training courses is really geared towards people going in industry and seasoned industry professionals. So we have a lot of customized training programs. They are run on both a contract basis and a customized basis. And again, we cover the gamut from gene therapy, mRNA vaccines, traditional recombinant processes, and we train people all over the globe. So with that, let's go ahead and get into the meat of today's presentation. We're going to be just focusing, again, high-level gene therapy manufacturing process. So we'll start by kind of defining exactly what gene therapy is. We'll get into kind of gene therapy vectors. So since we are talking about genetic modifications, what exactly is a vector and how are those used? And then the last thing that we're going to talk about is gene therapy manufacturing processes. So we're going to go ahead and cover NCTM's in-house AAV2 manufacturing process, but we're going to kind of talk about it in general terms to be all-encompassing of other platforms as well. So I always like to start my presentations with the drug product life cycle. And so this is kind of the pathway that drugs will go through from discovery all the way through commercialization. What I really wanted to highlight here is that the whole life cycle is very complex and there are a lot of activities that are kind of going on in parallel. So a molecule or a gene therapy product will start in discovery. So that's where our lab scientists really are trying to come up with new therapeutic molecules. They'll go through some process and analytical method development and then go through safety studies. So these are toxicology-like studies just to make sure that these products are safe before we bring them into people. So assume that the safety studies look good. A company will file an IND application with the FDA. And basically what this is it's an application in order to have a company be approved to start clinical trials. If the FDA approves that IND application, the clinical lot manufacturing can begin, and then the clinical trials will be conducted. So clinical trials are kind of split into 3 phases. Phase 1 is really just looking at safety. Is this product safe? Phase 2 starts to look at efficacy. So it will be expanded to patients that actually are suffering from the disease that the molecule is trying to target. And then if the phase 2 data looks good, the phase 3 clinical trials are then expanded to larger populations. Again, you're still looking at safety, you're still looking at efficacy, and now we're starting to kind of hone in on dosing and things like that. So at the conclusion of all your clinical trials, if all the data looks good, you can submit a BLA, a biologics license application, to the FDA. And that basically is what the FDA will approve before they will give you a license to actually sell that product. So while all of those clinical trials are going on, there are a lot of other activities that are going on in parallel. So while you're trying to figure out if the product is safe and if it works, you want to make sure that you are prepared if you do get granted a license, that you're ready to have material hit the market immediately, just because the patent lifetimes are defined and they're limited. And so you want to really maximize that time that you have to have that control over your market share. So while clinical trials are going on, you're going to be doing a lot of process development to kind of optimize your manufacturing procedure. That process development studies will give you information about what your facility needs to look like in order to support that manufacturing process. So a lot of times for new products, facilities will be designed and constructed specifically for new products. When you're constructing a new facility, it's not just building a building, it's also equipping that building with all of your equipment. So procurement and commissioning, qualification. So every time you bring a piece of new equipment into your facility, you need to go through IQ, OQ, PQ. Once your equipment is in-house, you'll be doing process validation runs. So your PPQ runs will be done in order to make sure that you have a really good handle on your process and that it's well controlled and defined. If you're using hard pipe systems, as part of that, process validation, you'll have to be doing cleaning validation studies as well. And that can be time consuming as well as your analytical method validation. So once your product, once you have your BLA submitted, it's approved, you have your facility ready to produce your product, you can start commercial production. Before any batches of that can be released to your patients, you have to go through quality control testing. So at this point, you've done a ton of work, and finally you have product that is making its way to customer. Now your work is still not complete at this point. You'll still have to do post-marketing surveillance. And so we kind of refer to this as phase 4 of the process, where you're constantly getting feedback from customers or patients on how that product is performing. And this is how we get information for if we need to do any modifications to recommendations for when that product is going to be used, if there are any recalls that need to be issued. All of that is informed by kind of this post-marketing surveillance. So you can see that the drug product life cycle is very long and complicated, and there are all of these different activities that are going on kind of in parallel. And this isn't just specific for gene therapy products, this is just kind of biologics as a whole. So gene therapies, generally defined, are a technique that modifies a person's genes to treat or cure disease. So there are different ways that we can do this. We can either replace a disease-causing gene and replace that with a healthy copy of the gene. If you have a gene in a patient that is maybe upregulated unintentionally or causing other issues, you can try to inactivate a disease-causing gene. And then the last way that you can approach gene therapy is by introducing a new or modified gene into the body to help treat a disease. So there are different ways that you can approach gene therapy. So all of these different ways rely on us introducing genetic material into a patient. So new genetic material into a patient, and there are different ways that we can do that. The most common way that we're approaching this is by using viral vectors in order to introduce these new genes into patient cells. There are other ways that you can get these genes into patients that are listed here, but we're going to focus on viral vectors for this presentation. So when you have these vectors that are essentially delivering new genes to a patient, there are different ways that you can approach that as well. So there's in vivo gene therapy, which is where you are basically taking your gene therapy product and injecting it into a patient. There's also ex vivo gene therapy. And in ex vivo gene therapy, what you're doing is you're basically taking cells from a patient, you are genetically modifying them in the lab, and then you are reintroducing those patient cells back to them. So a lot of our current CAR T-based therapies or autologous cell therapies are this ex vivo type of gene therapy. So just a brief background on the history of gene therapy. We've been doing work in this field for the last 30 plus years. Where we really hit a roadblock, at least in the United States, was in 1999, where during a clinical trial, Jesse Gelsinger actually passed away due to unfortunate immune reaction to one of the viral vectors that was doing or delivering the gene therapy to him. And so that really pumped the brakes a lot on the gene therapy field, at least within the United States. Meanwhile, China and Europe started licensing products, and the US ended up finally getting more on board with the gene therapies and approved its first gene therapy product in 2015. And to date, we have a handful of products that are licensed and on the market. So the ones that are licensed, at least in the United States, are listed here. They are a combination of both in vivo and ex vivo therapies, and they're going to be using some different viral vectors in order to transfer those genes to the patients. Now, the pipeline for gene therapy products is very extensive. So these numbers are a few years old, but you can see that there are thousands of products that are in the pipeline for gene therapy worldwide. And that number just continues to grow as more and more companies and more investors become interested in this space. And really the big driving force for gene therapy as a whole is that it actually has opened up the door to curing genetic diseases that we weren't able to before. So by being able to really change a patient's genes or their genetic expression of genes, we're really able to target a lot of new disease areas that we weren't able to before. So in terms of gene therapy vectors, and before I go any further, I just wanted to clarify that the word vector in this context really is just referring to a delivery mechanism. It's not referring to like a plasmid. Sometimes plasmids are referred to as vectors. That's not how this word is being used in this context. Vector just really means a delivery mechanism. So we can use viral-based vectors to deliver genes to patients. We can use non-viral-based vectors as well. So again, in the context of this presentation, we're going to focus primarily on the viral vectors. So a comparison between viral vectors and non-viral vectors. Viral vectors tend to be a little bit more preferred just due to the reasons on this slide. Generally, you're able to get higher titers for viral vectors compared to non-viral vectors. Long-term effects, they tend to be a little bit safer in terms of immune reactions as well. Although some of the non-viral reactors can also display this low immunogenicity profile. But I guess the take-home message from this is that no single vector is going to be suitable for all applications. And so when you're doing your initial experiments in the lab, it's really important to kind of look at different delivery mechanisms or different vectors to figure out which one is going to be best for your specific therapy. So before we go too much further into our discussion on viral vectors, I thought it would be important to kind of define exactly what viruses are, since a lot of these viral delivery mechanisms are based off of viruses. So viral particles are very simple in the grand scheme of things. Essentially what you have is you have some sort of nucleic acids. Those are going to be encapsulated in a protein coat, which is called capsid. And some viruses will also have an envelope of lipids that surround the capsids. So the way gene therapy works is that these viral vectors are modified so that your gene of interest is encapsulated within these viral particles. These viral particles are delivered to cells, and the cells basically uptake them into the endosome. The capsids or the viral particles will make their way towards the nucleus. Within the nucleus, the genetic material that is being enclosed by the vectors will be released. That DNA can then be transcribed into mRNA, and then from mRNA, we can get protein translation for whatever our target is that is encoded by those genes. Some things that are going to be important when we are determining or designing our viral gene therapies. The biggest thing is going to be target cell specificity. So you don't want to inject a patient with viral vectors and not know exactly where it's going to go. Usually for most diseases, you have a specific target that you want those vectors to be delivering to. You want to make sure that that delivery is efficient. And you want to make sure that ultimately you're not causing an immune reaction within a patient. So there are three commonly used viral vectors. There's AAV, which is adeno-associated virus. There are adenoviruses, and then there's lentiviruses. The product that was used in the Jesse Gelsinger case was an adenovirus, and generally adenoviruses tend to have a little bit higher immunogenicity compared to AAV or lenti, so usually it's not used as much compared to those two. Between AAV and lenti, there are a few different differences there as well. The one big one is packaging capacity. So how large is the gene that you're able to package inside of these vectors? AAV packaging capacity is significantly smaller compared to lenti. So depending on the size of the gene that you need to transplant, that is going to drive some of your decisions on which vector you're using. AAV, generally speaking, though, has the lowest immunogenicity across the three. A little bit more information about AAV vectors. So they are the smallest gene therapy vector. There are 11 serotypes, and where this is going to be important is on this next slide when you're talking about target cell specificity. So the really nice thing about AAV is that the different serotypes of AAV specifically target certain types of cells. So for example, if you had a therapy that you wanted to target in the kidney, you would choose, for example, AAV2, and that would target more kidney cells compared to other parts of the body. You can see here that the different serotypes, again, are a little bit more prone to delivering to certain types of cells. So some of the risks associated with gene therapy, obviously the big one is unwanted immune system reaction that can be very severe and very serious. Other problems can be, targeting the wrong cells. For example, you don't want to be modifying healthy cells, for example, and damaging them. If you are inserting genes, you don't want to have them going in the wrong places. There's always the risk of tumor formation and other side effects as well. So these are some of the things that we need to be cognizant about when we are designing our therapies. So processes, how do we actually make these gene therapy products? That's going to be the next section of our talk this morning. So again, there are different types of gene therapy products. There are in vivo products and there are ex vivo products. The in vivo products are the ones that you're going to be injecting into patients. And so they look very similar to the products that you'll see for, general biologics or vaccines. They'll be usually in vials that will then be taken up into a syringe and then injected to a patient. Your ex vivo therapies look a little bit different. So these are the ones where you're actually taking cells from a patient, you're genetically modifying them in the lab, and then you're reintroducing them. So this pouch here on the right, which is a picture of Chimera, that's actually filled with modified patient cells, which will then be transfused back into a patient during administration. So for the in vivo therapies, the components within that vial are mostly going to be your active molecule excipients to help stabilize that product while it's in storage and give it some measurable amount of shelf life. And then there are going to be trace amounts of process and product related impurities. And in terms of our impurity profile, obviously the fewer is better. And as we go through kind of our manufacturing process, we'll talk about what kind of impurities may be there and how we can make sure that those are minimized as much as possible. But the majority of what's in that vial is actually water, and it's usually a high purity water, water for injection. So here's just some information. It's taken straight from the drug packaging insert from Luxturna. Again, you can see that this comes as a concentrated drug that will be diluted before administration. But you have your viral vectors, and then just a few excipients and a lot of water. So again, your AAV dose is going to be dependent on the route of administration. So if you have an in vivo therapy that you are injecting into a patient, generally your doses are going to be rather small compared to if your dose is delivered systemically. So again, a lot of those CAR T cell therapies or whatnot are going to require much higher concentrations of vectors per dose. So that's going to directly lead into manufacturing considerations when you're trying to scale your facility. Another thing that's going to be important when you're determining the scale of your facility is the disease that you are targeting. So some of the diseases in the gene therapy space are rare diseases, and there are less than 1,000 patients that are going to be treated per year. Obviously, in those situations, you don't need a huge facility that's running 24/7. You probably might need one batch, if that, per year, compared to a therapy that's targeting something like Parkinson's that has a lot more patients per year. So making sure that you do the math and calculating exactly what size batches and how many runs per year you need to do in order to meet the demands of the market and your patients are going to be really important when you're doing those scaling exercises and figuring out what size batches you need to run. So the overall manufacturing process is still rather new in the grand scheme of things. I feel like we have a lot of very well-defined templates and platforms for traditional biologics, but with gene therapies, we've kind of borrowed some of the best practices from biologics, but we're still kind of figuring out exactly what that platform is for this new space. But ultimately, one of the things that we want to make sure that we're doing is that whatever unit operations we're choosing to use, that they're scalable. It doesn't do us any good if we've designed this perfect process that can only be, utilized on the microliter scale. That's not going to be feasible for commercial manufacturing. And so we just need to make sure that, again, the unit operations that we choose to use in our process are scalable. We'll talk about some of the scalable unit operations in the next several slides. So generally speaking, again, we can kind of divide up our manufacturing process into upstream and downstream. Again, very similar to our traditional biomanufacturing processes. So in upstream, you're essentially growing up your cells and producing all your viral vectors. And then once you get into downstream, you're going to be getting rid of all the impurities and making sure that what you have left at the end of the day is a product that is safe and efficacious for our patients. So the downstream process can be split into 3 blocks. The first block is going to be your harvest operations, where you're really just doing a phase separation between your cells or cell debris and your spent media. Then you'll go into the next stage, which is purification, where you're really trying to remove all of those impurities that we talked about earlier. And then lastly, in stage 4, we're going to be formulating and filling our products. So you want to, at that point, your goal is to essentially get your product at the appropriate concentration that you need for dosing purposes, as well as in a formulation that will promote long-term stability and storage for that product so that you have some time before you are required to use it for a patient. Here are some of the unit operations that we use in our in-house AAV2 production process. So we use, your traditional bioreactors for our upstream operations. For our downstream operations, we're using a combination of depth filtration and ultrafiltration. Before we go into our chromatography unit operations for purification. And then lastly, for formulation, we're using ultrafiltration and then our bulk filling units. So a little bit more detail about the different stages. So stage 1, again, is your upstream production using either a HEC 293 or an insect cell line SF9 in order to produce our AAV2 vectors. We use a triple transfection process. So we have 3 plasmids that are being transfected. We have our plasmid with our gene of interest. For us, it's GFP. Since we're a teaching organization, we use that as kind of our teaching tool. We also have a construct with rep and cap genes, and then the third plasmid is our helper plasmid. So when you are doing your upstream process development, you're going to want to optimize your transfection conditions. So there are a lot of different parameters that you can optimize. And once you have kind of your optimization process figured out, you'll also want to make your working cell bank and your master cell bank similar to what you would be doing for a traditional biologic. Your process development or your production train for your culture, again, follows a very similar path that you would be using for a traditional biologic. So you'd start with a vial from your working cell bank. You would go ahead and scale that up into larger and larger bioreactors until you get to your production bioreactor. It's important to note that through all of this scale up and subsequent inoculations, that you're only growing up either your SF9 cells or your HEC 293 cells in those reactors. Once you get to your production bioreactor, again, you're still growing up those cells. You want them to be healthy. And then once you get to your optimal density, you will then transfect those with a cocktail virus. So it's only in your production bioreactor that you're actually doing that triple transfection. What happens at that point is those viruses go in and start infecting the healthy cells in your reactor, making more copies of themselves. And it's through this infection process that you're able to actually reproduce those viral particles and make your viral vectors that is ultimately your product. At the end of that infection, you will go ahead and harvest the material, and you'll have to basically start by separating out your viral vectors from all of the other junk that you've produced to this point. There's some pictures of bioreactors. So there are both single-use options like the wave bag on the left and hard pipe systems like the stainless steel systems shown on your right. And either of those are going to be viable options for this process. The main objectives when you're doing your bioreactor design, obviously you need to maintain aseptic operation and you need to maintain, your basic aeration, agitation in order to meet metabolic requirements of your cells. So going into our downstream processes, harvest operations are going to be next. A lot of times we will be, actually, let me go back just a second. A lot of times we'll be actually lysing those cells within the bioreactor. Sometimes, depending on the serotype of AAV that you're using, sometimes the viral vectors will be secreted into the media. Sometimes they are going to be produced and stay within the cells. So for AAV2, most of that expression happens intracellularly. So we actually have a cell lysis step that we use, and we actually lyse directly in the bioreactor. After that lysis step is completed, we send our material over a depth filter in order to separate out all of the broken up cells and separate it from the media, which contains our AAV particles. So you can use a number of different unit operations in order to achieve this lysis and subsequent clarification step. For clarification, you can use a depth filter like what's shown in B here. You can also use centrifugation. So what's shown in figure A is a distack centrifuge, which can also be used to separate out the solid cell debris from the liquid that contains your viral vectors. In terms of lysis, you can use a homogenizer, which is shown in part C, which is basically a mechanical way to break open your cells. There are also surfactant-based ways that you can do cell lysis as well. So just some examples for different harvest trains that you might investigate. So train 1 has you lysing your cells in the bioreactor. That is followed by a centrifugation step, which is then followed by a secondary depth filtration. And then finally, you go through a 0.2 micron surface filter before you go into your next unit operations. You could also replace that centrifuge with a series of depth filters. So you can have a primary depth filter followed by a secondary depth filter before you go onto that surface filter. One last method that you can look at is using tangential flow microfiltration in order to run that separation step. Ultimately, what you want to do is you want to make sure that you're removing as much of the solid as possible before you get to that surface filter at the end. It's because that surface filter is going to have very low solids capacity. And so you want to make sure that you're removing, again, as much of that solids that's possible before you get there. So we talked earlier about how sometimes you could use a surfactant in order to drive lysis compared to a homogenizer. So if you are using a surfactant-based lysis step, there are a lot of different process parameters, such as the ones listed here, that you can investigate for that process optimization. One thing that is really nice about lysis using surfactants is that this is going to be compatible with single-use process trains. So you can mix the surfactant with your material in a single-use mixer or even within your bioreactor, if it's a single-use bioreactor, as long as you can make sure that those mixing conditions are sufficient. And then you can go ahead and really implement this in a single-use process train. If you're using something like a homogenizer to drive your lysis step, that is unfortunately not going to be compatible with a single-use process train. So depending on what is important to you will also help determine kind of which unit operation you're going to choose for your particular process. So distack centrifugation is another method that you can use for clarification. This is just a generic cartoon diagram that shows how a distack centrifuge works. Essentially, you have your feed, which contains a mixture of your solid cell debris and your liquid containing your viral vectors. That is spinning, and so the solid cell debris is going to be more dense than the liquid, so it kind of accumulates on the outside walls of that bowl. And that can be intermittently discharged while the clarified liquid continues to flow through that centrifuge, and you're able to perform this kind of solid liquid separation in a continuous fashion. This is what a large-scale centrifuge looks like. And again, in order to operate this, it's a fairly straightforward process. And essentially, you're just going to be continually feeding the centrifuge until all of your feed material is exhausted. An alternative to centrifugation is depth filtration. So depth filtration is just a filter. And the special thing about depth filters is that they are very high solids capacity compared to your surface filters, which are very low solids capacity. So essentially what you have is you're feeding your material, which again is this mixture of solids and liquids, that will get fed through your depth filter. The depth filter will capture all of those solid particles, and the clarified liquid will flow through that filter and can be collected. Once you get into stage 3, we're in our purification unit operations. We are going to be really trying to separate out our AAV from all of our other impurities. And there are going to be a lot of impurities because we've lysed open cells, and all of their host cell proteins and host cell DNA and all of these other impurities are going to be released into that liquid stream in addition to our AAV. So we're going to be using chromatography in order to run the separation process. This is what a chromatography skid looks like on the left, and the picture on the right shows you what a packed chromatography column looks like. And these are very much pilot scale columns. The large manufacturing scale columns can go up to 2 meters in diameter. So when you're talking about purification, it's important to define exactly what your impurities are so that you can talk about what it is that you're trying to remove. So there are 3 different types of impurities. There are process-related impurities, so that's going to be impurities that are derived from your manufacturing process. So your host cell DNA, host cell proteins, endotoxin, all of these things would be examples of process-related impurities. You also have product-related impurities. So these are going to be variants of your product. So anytime you don't have a properly folded product, that becomes an impurity. So even if it's very, very similar to your actual target vector, if it's misfolded or modified in any way, that becomes a product-related impurity. And then lastly, you have contaminants. So these are external components that are introduced into the process that are not actually part of your process. So generally speaking, chromatography is going to rely on the use of a solid support that's going to interact with either the impurities in your mixture or your product while other things flow through. And so you are going to have a stationary phase, which can be a resin, it can be a membrane or a monolith, and that stays stationary, hence the name. And then you're going to be flowing liquid over that stationary phase. So the liquid is referred to as your mobile phase. So the way you get separation within a chromatography column is kind of as illustrated in this cartoon. So you have a mixture of different components that are loaded to the top of your column. And each of the different components in that mixture are going to interact with that solid, the stationary phase at different levels. So here, which we have a mixture of 3 different components, a blue, a green, and a red species. And the blue component is very much attracted to the resin. The red component is not attracted to the resin at all. And so because the blue one is very attracted to the resin, it really wants to interact strongly with the resin. So what that means is it's going to take its time as it moves through the column. It really wants to bind to that column and it doesn't really want to move. Whereas the red component here isn't attracted to the resin at all. So it's just kind of freely flowing through the column. And so the different rates of speed at which these different components are traveling through the column generally lends itself to separation. So if you collect the stuff coming out of the bottom of the column, you can see that the red material comes out first, followed by the green, followed by the blue, and they can be collected separately. And that's essentially how you're able to drive the separation of mixtures. So obviously in your process, you're going to have many more than 3 components, but the general gist is still kind of the same. There are different types of chromatography resins out there that are going to interact with components in different fashions. So there are ion exchange resins. Those are going to be interacting with different solutes based on differences in charge. You have affinity resins, which are going to be interacting with different components in a very specific biomolecular interaction way. So there are affinity resins, for example, that will specifically bind to certain serotypes of AAV, and it will only bind to certain serotypes of AAV, and everything else will flow through. So affinity resins are really powerful in that you're able to get really high purities because that interaction is so specific, just based off of a single column step. Hydrophobic interaction chromatography relies on differences in hydrophobicity in order to bind components. Reverse phase is also another hydrophobic interaction, but that is a very strong interaction. And so usually that is used in the QC lab and not necessarily for preparative methods. Size exclusion is similar. Size exclusion is separating based on size. And then you also have mixed modal resins that will use a combination of, ion exchange and reverse and HIC, for example, that will give even a different level of separation. Lastly, in stage 4, formulation, we're going to usually be using ultrafiltration skids in order to drive the formulation of our products. So here's a picture of an ultrafiltration skid, and the skids are always going to be utilizing ultrafiltration membranes. The membrane is actually where all of the separation is occurring. So very generally speaking, ultrafiltration is a membrane-based process. So it's a size-based separation. And the way that it's designed is that you want to choose a pore size in this membrane that is smaller than your product. So you want that membrane to be retaining all of your viral vectors. Anything that's going through is basically going to waste. And so what that allows us to do is it allows us to both concentrate our AAV, and it's also going to allow us to do a buffer exchange to change out whatever our AAV is dissolved in. And that's going to be important for our formulation operations. So typically what you're going to do is you're going to first start by concentrating your product. A lot of times the concentrations that we're working with in purification are going to be fairly dilute compared to the concentrations we want our final product to be on. So we're going to start by concentrating. So we're going to essentially start recirculating our material. Material from the feed tank is going to be pumped to our membrane. Our membrane is represented by that dotted line inside the rectangle. So remember, our AAV is not going to pass through that membrane. It's going to come out in the retentate stream. The retentate stream is going to be redirected back to that feed tank. So our AAV is basically going around and around in that recirculation loop. The permeate stream, the stuff that does go through the membrane, is going to be your water and small buffer components, anything that's small enough to fit through the pores of that membrane. And so essentially, as you turn on your pump, you're sending permeate to drain, which is ultimately causing the level in your feed tank to decrease over time. Remember, your AAV is just stuck in that recirculation loop. So your concentration of AAV is increasing over time while the volume in your feed tank is decreasing over time. After that, you can go ahead and formulate your product. So we use the term diafiltration to refer to our buffer exchange step. So essentially what you're doing here is you're hooking up your diafiltration or your formulation buffer to a pump that's going to feed that feed tank at the same flow rate as that permeate stream is leaving. And so you're essentially setting it up like a steady state system. And so once you turn on your pumps, you have diafiltration or formulation buffer going into your feed tank. Permeate is still going to drain. The concentration of your AAV and the volume in your feed tank are remaining the same, but the composition of what that AAV is dissolved in is what is changing over time. And that's going to allow you to formulate your product into whatever solution you need for patient administration or long-term storage. So I'm going to skip over the math on this slide, but basically this is doing a mass balance on the old buffer remaining in your solution. Because if you plot the results of that differential equation, you essentially get a graph that looks something like this. And what this is going to tell you is it's going to tell you exactly how many diavolumes. So a diavolume is referred to as whatever volume is left in your feed tank. How many diavolumes of buffer exchange you need to execute in order to get a certain amount of impurity removal or formulation. So typically for a final formulation, you're going to be somewhere in the realm of 7 to 12 diavolumes, just in order to make sure that all of your processing chemicals are removed to a very high extent and really only trace levels of impurities are left. So again, going back to our discussion about scalable versus non-scalable unit operations, we want to make sure, again, that all of the unit operations that we're choosing are scalable. So for example, bottle centrifuges are not scalable, but disc stack centrifuges, for example, that can afford us continuous flow, that is something that's scalable. Another example of this is doing buffer exchange. So an example of a not scalable unit operation would be something like dialysis, whereas a TFF or an ultrafiltration system is an example of something that's scalable. So just keep in mind, especially if you're early on in your process, the earlier you start using these unit operations that are scalable, the less development efforts are going to be needed later on. Now, there's been a lot of interest in the use of single-use processing and single-use components within processing, and there are a lot of reasons for that. So first of all, speed is always going to be very important to us. Speed to market is a very, very important thing for manufacturers, and using single-use equipment does help with that speed to market because it primarily helps you with the speed because it eliminates cleaning validation altogether. And so you're able to save a lot of time on those efforts. You're also able to usually turn around equipment faster because you don't have to clean and sanitize anything. You just throw away the old flow path and get a new flow path or bag or whatever it is that you're using. And that reduces your turnaround time for equipment. In terms of capital costs for new facilities, generally speaking, you have lower capital costs because you're essentially just buying the shell of an equipment, and you are going to be lining that shell with, your single-use components compared to having to buy large stainless steel pieces of equipment. It just ends up being a lot cheaper for your initial equipping of your facility. However, with single-use equipment, you will have to be buying those consumables, obviously. Some of the reasons why single-use technology is still, being implemented in certain areas, some of the unit operations, so for example, homogenization, don't have single-use options. And so not all of the unit operations as of right now are going to have single-use options. Again, when we're talking about lysis, you can switch to chemical-based lysis procedure if you wanted, but some of the unit operations, again, are a little bit more amenable to adaptation to single use compared to others. It's going to increase your reliance on outside vendors. So I think with COVID, it really gave us good insight in terms of the impact of supply chain and how that can impact all of our operations. And then lastly, on the QC side, obviously for release testing, there are going to be a lot of assays that are going to be required for your gene therapy products. And just for the sake of time, I'm not going to go into these in detail, but you're going to have a whole set of release criteria before those products can be released. Lastly, just wanted to reiterate the importance of operating under cGMP procedures. And for most of our manufacturers, this is something that they're already doing as well. Again, cGMP stands for Current Good Manufacturing Practice, and it's something that we really stress at NCTM when we're training all of our students going through our programs. Enforcing GMP is going to be really important, especially just bottom line safety of all of our patients. And cGMP is just this huge overarching process that is going to impact all of the different aspects of your facility and your operations. Again, all of these different components will come together under cGMP, ultimately to just ensure that the products that are coming out of our facilities are quality and safe. So if you have any questions, feel free to reach out, or if you'd like to learn more about our programs, feel free to reach out, and I'd be happy to take any questions.
Thank you, Dr. Reeves, for that wonderful presentation. We will now start the live Q&A portion of the webinar. If you have any questions you want to ask, please do so now. Just click on that Ask a Question box located at the far left of your screen. So let's get started. It looks like we already have a few questions coming in from our audience. Our first question, can you elaborate on your hands-on gene therapy manufacturing in-person course?
Sure, yes. So the presentation that we just went over is just kind of our introductory lecture for an actual week-long hands-on manufacturing course specific for gene therapy products. So our courses are small group format, so we cap participants at 12 per cohort just to make sure that we really give students ample hands-on time with the equipment. And we also do a lot of redundant equipment. So for example, when we do ultrafiltration, for example, we actually have 4 groups running at the same time. So we really make sure that the hands-on piece is really central to our training programs. So if you went through one of our training classes in person, we would basically take you through the entire process, starting from upstream. So we'll have you grow up either HEK 293 cells or SF9 cells. You'll do the triple transfection in the lab, grow up those cells, you'll infect them. And then we'll go through the whole downstream process where you'll be harvesting that material with the depth filter. Then you'll go ahead and purify your AAV on chromatography, and then we'll formulate it with ultrafiltration. So the courses are very much hands-on. Like I said, they're about 65% lab-based and then 35% lecture. So we'll usually use the lecture time to kind of introduce a concept, and then we'll go into lab and actually execute the concepts. And then in terms of the format of the labs, they range in scale from kind of microliter scale all the way to more pilot scale equipment that we're using. So it's very much relevant for our industry partners.
Wonderful. Thank you so much. And our next question, what is the outlook on single-use consumables in this field, and how are the environmental concerns addressed?
That's a really great question. Single use is definitely becoming more and more prevalent in the field, and it's for a variety of reasons. We touched upon a few of them in the presentation about, decreasing the risks of contamination, which is really critical, especially for like our cell therapy products. And then, decreased time for turnaround and the elimination of cleaning validation. In terms of the environmental considerations of single-use products, it's a tricky one to address. On one hand, single use is actually less carbon intensive than some of our hard pipe systems. When you take into account the amount of clean water that's required to clean and that's required to support your cleaning validation studies, all that high purity water, so your WIFI and things like that, that's very carbon intensive in order to produce. And so with the elimination of cleaning and all of the associated studies revolving around that, it actually comes out fairly carbon neutral between, cleaning with all of that high purity water and then the environmental impacts of the plastics and the disposal of those plastics. In terms of recycling of the bags, it's a really tricky area to get into. A lot of our single-use components are actually multi-composite films, which makes recycling them very tricky because it's hard to kind of extract the different components. I know there are some solution providers that are looking into new films and recyclable films. So I think Sartorius, for example, is working into that field, and they're not the only ones. But as of right now, it seems like incineration of the bags is might be the best way to go in terms of like being able to recover some of the energy from that incineration process. But it's not a perfect solution, and I hope that we see more green alternatives in the future.
Thank you so much. And it looks like we have one more question coming in from our audience members. What does it mean to be simulated cGMP facility?
So NCTM is a simulated cGMP facility, and what that means is that we try to be as GMP-like as possible. So when we are training our students or the people that are going through our programs, we really try to operate in a cGMP fashion, and that looks like, it could look like a number of ways. So for example, our facility is designed to be simulated cGMP, so we have gowning suites, we have unidirectional flow, we have airlocks going in between suites. And then we also have our students operate or execute all of their protocols using written protocols, written procedures, which is compatible with, a true cGMP facility. So all of our labs are run off of SOPs and batch records, for example. We also teach GMP concepts like documentation, qualification, validation, things like that. And so that students are really getting a good flavor for what goes on in a production environment.
Dr. Reeves, I want to thank you for your presentation today. Do you have any final comments for our audience before we go?
I don't think so. I thank you all for attending, and if you have any questions, feel free to reach out.
Well, thank you again, and thank you for your important research. And before we go, I want to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed for 2 years until April 5th, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.